Hi, our topic was protein synthesis, and I will be doing a general overview of protein synthesis, and then they'll go into more detail. So this is basically the nucleus, and these twisters represent the boundaries of the nucleus, and this is DNA, and this is the cytoplasm. So basically, the promoter on the DNA calls for the RNA polymerase, and then the helicase starts breaking the hydrogen bonds, and then the RNA polymerase gets the free-floating nucleotides, and then attaches, forms hydrogen bonds, and then mRNA is formed, like here, and then the rRNA gets the mRNA, and then the tRNA starts to form bonds with the mRNA, and then let's see. And then amino acids are attached to the tRNA, and then polypeptide bonds are formed with other tRNA. Other tRNA also attach to the mRNA and create polypeptides.
Okay, so to go more into transcription, what it is basically taking the genetic information of DNA and remaking it into RNA. And so that happens in the nucleus, and what happens is you first have the original DNA, and it's in a double helix form. And so what happens is the RNA polymerase comes and it unwinds the DNA like this, and that, and then the hydrogen bonds are broken by the helicase, so it starts to split. And then, so what happens is basically transcription happens in 3 steps. So in the first step, the RNA polymerase is attached to a nucleotide that has a specific sequence that is called a promoter, and the area around it is called the promoter region. And so the promoter region contains 3 sections that is called the regular site, the tautai site, and the initiation site. And so the basically where the promoter is in the initiation site. And so since RNA polymerase can't do all the job by itself, it has helper proteins that help, and there is a protein called the sigma factor that attaches to the RNA polymerase. And so the helper proteins are attached to the tautai site and the regular site, and the RNA polymerase itself is attached to the initiation site. So what happens is, in the second step, the RNA polymerase goes down the strand and matches the corresponding nucleotide to make the RNA. And so in RNA, instead of thymine, there's uracil that goes with adenine, and similar to DNA, cytosine goes with guanine. And so in the third step, what happens is it goes down the line and it reaches a specific nucleotide called the terminal termination signal, and that's where it tells the RNA to stop and to get off the RNA to be used once again in another DNA. And so after that, after the RNA polymerase is gone, the RNA splits from the strand of DNA. Oops. It's split from the strand, oh gosh.
So it begins to split, and this is in the cytoplasm. So after this part of the mRNA is split from this, it bonds a ribosomal rRNA, helps to bond the tRNAs with this, like so. And this is methionine, this is glycine, this is serine, and this is valine. And this is the stop codon, which is UGG, and this is the start codon, which is AUG. And these twizzlers represent the codons. So this is these 3 are codon, these 3 are codon, these 3, these 3, these 3. And then basically, okay, so then peptide bonds form between these amino acids like this, and then they would fold up to make a protein, like so.
Okay, so this is a chart of all the possible mRNA codons, and genetic code is the correlation between nucleotides and the amino acids that they code for. And if there are 3 nucleotides, it's a codon, and each codon codes for 1 amino acid. However, different codons, for example, this one and this one can code for the same amino acid. So these are the codons that have the first nucleotide U. These are the codons with the first nucleotide C and A and G. And these are the stop codons. They are UAA, UAG, and UGA. And these are the stop codons, UAA, UAG, and UGA. And this is the start codon, which is AUG. And the start codon signals for the start of translation, and the stop codons signal for the end of translation.
If I may add in something, so after the RNA is made, it doesn't always have to be made so that it is part of the translation cycle. It can either be made into tRNA, which is transfer RNA, or it can be made into which it's an original mRNA, which is just like a strand, or it can be made into rRNA, which is part of the ribosome.